Hello. We're going to focus this discussion on pediatrics and how this applies to your prescribing and how it affects pharmacokinetics, dynamics, and pharmacotherapeutics. So let's get started here. We're going to start off with a little background and talk a little bit about the regulation and the legal oversight that goes into prescribing for the pediatric population. So let's start off with talking about the federal drug regulation. Now, drug regulation in the United States has often been moved forward after tragedies have occurred that directly involve children. Regulation began with the passage of the Federal Food and Drug Act of 1906, which was enacted because children had died from ingesting tainted food products and soldiers had died from ingesting adulterated quinine. This law, was known as the Wiley Act, prohibited the manufacture and interstate shipment of adulterated and misbranded foods and drugs. In 1938, the Federal Food, Drug, and Cosmetic Act was the next major legislation enacted. It was passed as a result of continued adulteration of products including sulfonylidamide, which has caused more than 100 deaths in children because of diethylene glycol used in the elixir. For the first time, documentation of drug safety was mandated. This act also mandated truthful labeling and established the new drug application process that required toxicology testing prior to drugs being promoted and distributed. In 1962, the Harris-Kefauver Amendment was passed, prompted by the birth of thousands of deformed infants whose mothers had taken the sedative thalidomide. This amendment mandated preclinical animal trials before testing drugs in humans. It also established 3 phases of clinical testing. Phase 1 establishes safety and pharmacokinetics. Phase 2 establishes initial effectiveness and dose range. Phase 3 conducts comparative clinical trials. Although the Harris-Kefauver Amendment increased the safety of new drugs coming into the market, it also slowed new drug development, increasing the time from an investigational new drug to the new drug approval to 8 or 9 years. In 1972, an over-the-counter drug review process was begun to enhance the safety and labeling of non-prescription medications. In 1986, the Child Vaccine Act was passed requiring patients and parents to be informed regarding the vaccines they are being given. In spite of all these laws, as late as the 1990s, the percentage of approved drugs that contained no labeling information for children was approximately 70%. The next major legislation that affected children was the U.S. Food and Drug Administration, FDA, Modernization Act of 1997. The act had 2 main components pertaining to pediatrics. 1, the FDA could require in writing that the manufacturer submit data on pediatric patients for drugs that appear to have a pediatric use. Previously, drugs were approved for use in adults, and the pediatric prescribing information happened later. This law mandated that the FDA require the pediatric data up front. 2, pharmaceutical companies were rewarded by a 6-month extension on any patent if they voluntarily test the medications for safety in children. The FDA Modernization Act was challenged by the drug companies and overturned in 2002, which was a major setback for pediatric drug safety. Fortunately, the American Academy of Pediatrics and other groups concerned with pediatric drug safety went before Congress, and the result was the passage in 2003 of the Best Pharmaceuticals for Children Act, commonly referred to as the BPCA. This act reinstated the pediatric exclusivity rule, giving a 6-month extension on patents if a manufacturer studies a given drug in children. It also established a mandate for annual list of requested drugs to be studied. Experts in pediatrics are consulted and a list is developed for priority testing. In addition, the Pediatric Research Equity Act was signed into law in December of 2003. This act requires that all applications for new active ingredients, new indications, new dosage forms, new dosing regimens, and new routes of administration must contain a pediatric assessment unless the sponsor has obtained a waiver or deferral of pediatric studies. Another exciting development in pediatrics is an international movement towards improving pediatric drug safety. The The World Health Organization launched the Make Medicines Child-Size campaign in December 2007 to raise awareness and encourage medication development for children younger than age 12 years. A global alliance in pediatric pharmacology has been formed, and pediatric pharmacologists from more than 30 countries have met to share information from pharmacokinetic and pharmacodynamic studies and avoid unnecessary duplication of studies. This movement is very positive and encouraging, but you can see from the background how how much information is not available regarding medications in children. So next, let's move on, and we're going to look at the pharmacokinetics of drug therapy in infants and children. So pharmacokinetics includes drug absorption, metabolism, and excretion, and can vary throughout infancy and early childhood. Even at puberty, there are differences in drug clearance between girls and boys as drug clearance rates reach adult levels. As more is learned about the metabolic pathways in the adult liver, more knowledge is gained about how to study the differences in children. Past disasters caused by lack of understanding about the physiology of newborn metabolism have led to caution regarding the use of medications in infants. Gray baby syndrome caused by inadequate glucuronidation of chloramphenicol, which led to dangerous drug accumulation, and sulfonamide-induced kernicterus, caused by displacement of bilirubin from plasma proteins by sulfonamides, are two such disasters that have been hard lessons in the use of medication in newborns. Well-designed pharmacokinetic studies in the newborn and careful therapeutic drug monitoring have improved knowledge of neonatal pharmacology, yet care is essential when any new therapy is tried. Now let's look at drug absorption. Drug absorption can be affected in children more than it is in adults by three factors. Those three factors are: 1, the blood flow at the site of administration, such as with IM or sub-Q; 2, the gastrointestinal function; and 3, the thin stratum corneum. Now, neonates have more variability in the blood flow to the muscles, especially ill newborns, and poor blood flow can lead to delayed or variable absorption of medications. If perfusion suddenly improves, there can be rapid absorption of the medication from the muscle, leading to a possible toxic level. Care should be taken when administering potentially toxic drugs, IM, to ill infants. GI function is variable in neonates and young infants. Gastric acid function begins soon after birth and gradually increases over several hours. In premature infants, gastric acid secretion occurs more slowly and takes up to 4 days to reach normal levels. Gastric pH does not reach adult levels until 20 to 30 months. Gastric emptying time is prolonged and reach adult levels by 6 to 8 months, meaning medications absorbed from the stomach may therefore have increased absorption. The neonate also has slow and irregular peristalsis, and medications absorbed primarily from the small intestine should be monitored for potential toxic levels. The developmental changes in gastric function alter drug absorption in a fairly predictable manner. The oral bioavailability of acid labile compounds is increased, and the oral bioavailability of weak organic acids is decreased. Basic drugs such as diazepam and theophylline have increased absorption. Gastric motility greatly alters the absorption of drugs with limited water solubility. It is also well known that infants and young children have a thin stratum and larger body surface area in relation to size, and this affects topical absorption of medication. Children absorb topical medications more readily than do adults, leading to systemic toxicity seen with topical medication use. This is seen with topical use of lidocaine or diphenhydramine in young children. Most providers are familiar with the concern for systemic absorption of topical corticosteroids in children with systemic Cushingoid symptoms or hypothalamic pituitary adrenal axis suppression developing with topical use. Due to BPCA and pharmacokinetic studies that indicated HPA axis suppression, right? That is that hypothalamic pituitary adrenal suppression. In children, a number of topical corticosteroids have been relabeled. Now let's move on to distribution. There are clear changes in body composition in neonates, infants, children, and adolescents. Newborns have total body water of 80%, which drops over the first few months, to total body water of 60% at 6 months. Therefore, infants require higher doses of hydrophilic drugs. Infants also have a decreased volume of distribution for lipid-soluble drugs. Infants younger than 6 months have decreased plasma proteins available for drug binding, which will cause elevated levels of unbound medication. Providers need to monitor for drug toxicity even if they have normal or low plasma concentration of total drug. Phenytoin is one drug that this is seen in, as it is only 80 to 85% bound in infants and 94 to 98% bound in adults. The ratio of fat to lean muscle also shifts throughout childhood, with a shift toward decreased total body fat in adolescence, a shift of approximately 50% in males between ages 10 and 20 years. Consequently, lean body mass increases more in males. The shift in females is less dramatic, shifting from 28 to 25% from ages 10 to 20 years. Due to these changes, it may be difficult to predict pharmacokinetics of some drugs during pubertal growth. Medications that the patient takes chronically, such as seizure medications, need to be monitored closely during puberty growth. Now, metabolism. When we talk about metabolism, we're going to talk about also the phase 1 enzymes and the phase 2 enzymes. The pathways of drug metabolism develop variably over the first year of life and may be influenced by medications that induce drug metabolizing enzymes. In adults, much has been learned about the cytochrome P450 enzymes and much is still unknown. The exact developmental pattern is not known for most of the CYP450 isoenzymes, although our knowledge is increasing rapidly. Recent studies recognize the small intestine is a major site of drug metabolism because it contains enterocytes in the bowel mucosa, which have CYP450 drug metabolism enzymes. This information enhances our knowledge of drug metabolism, but there may be large inter-individual variation in the capacity of the small bowel to metabolize drugs. Studies of CYP450-1A2 using caffeine as the test substrate demonstrate limited metabolic clearance in the newborn, reaches adult levels at 4 months, and then exceeds adult levels at 1 to 2 years throughout childhood. At puberty, clearance begins to decline to adult levels in girls sooner than boys. Diseases such as cystic fibrosis can affect the CYP1A2 activity, and cystic fibrosis patients may need higher doses of medications that are metabolized via the CYP1A2 pathway. There are many medications that are metabolized via the CYP1A2 enzymes. Common ones include theophylline, erythromycin, cimetidine, clarithromycin, among others. Foods that are affected by the CYP1A2 pathway are grapefruit juice, cruciferous vegetables, and charbroiled foods. Cigarette smoking also affects CYP1A2 enzymes, and providers should inquire if patients taking medications are smoking, as it may affect therapeutic levels of medication, including a number of seizure medication. In practice, this implies that drug dosages need to be adjusted as a child goes through phases of CYP1A2 maturation. Higher dosages may be needed from 1 year until puberty, and therapeutic drug levels will need to be monitored as a child goes through puberty. The isoenzyme CYP3A4 is the most abundant CYP isoform and undergoes a similar maturation process to CYP1A2. CYP3A4 has low activity at birth and reaches 30 to 40% of adult levels by 1 month. By 6 months, it is at full adult level and exceeds adult level at 1 to 4 years. At puberty, it decreases to adult levels. The CYP3A4 enzymes are used to metabolize more than 20 commonly used pediatric medications, including prednisone, macrolides, antihistamines, oral contraceptives, and NSAIDs. The implications for pediatric practice include monitoring when prescribing more than one drug metabolized by CYP3A4 enzyme and monitoring during developmental changes. Now moving on, talking about phase 2 enzymes. Phase 2 enzymes are responsible for synthesis of water-soluble compounds. There is less information available on phase 2 activity in children. UDP, glucogeuronyltransferase, UGT is what we will call this, are responsible for hundreds of hydrophobic compounds. It is known that morphine is metabolized by UGT2B7. It is known from morphine studies in neonates that premature infants with a gestational age of 24 to 37 weeks have a much lower plasma clearance of morphine than children 1 to 16 years. It is thought that morphine clearance reaches adult levels at 2 to 6 months, although some children do not reach adult levels until 3 years. There appears to be ethnic variations in TPMT activity as well. Koreans do not reach adult activity levels until 7 to 9 years. Although little is known about phase 2 enzymes in children, but the knowledge base is growing. Commonly used medications such as acetaminophen, morphine, propofol, and caffeine are metabolized via the phase 2 enzymes, and providers need to be aware of developmental as well as possible ethnic variations. One essential consideration for knowledge is that during times of great physiological change, such as the premature, the neonate, puberty, there are likely to be major changes in pharmacokinetics. More variability among individuals and within the individual is likely during these periods. Careful monitoring of therapeutic drug levels is critical to safe outcomes. In the neonatal period, frequent adjustments may be necessary because of the rapid changes the neonate is undergoing. A drug dosage that is therapeutic level in a 9-year-old girl has to be carefully monitored as she proceeds through puberty to ensure that she will not develop toxic levels as her drug clearance reaches adult levels. Let's move on now to excretion. Drug excretion rates are affected by the lower glomerular filtration rate in newborns, which is only 30 to 40% of adult values. By age 6 to 12 months, the glomerular filtration rate, or GFR, reaches adult values. Drugs that depend on renal excretion are cleared more slowly in neonates. Drug dosages and dosing intervals in newborns are adjusted accordingly for medications such as ampicillin, aminoglycoside antibiotics, and digoxin. Renal blood flow is also reduced in neonates, and reaches adult levels at approximately 9 months. So we're going to take a break here. We've gone over the pharmacokinetics and some of the background. So from this, it's important to remember the names of the important acts and what the main outcome of the act was. Pharmacokinetics, clearly there are differences. Main points are that children need to be monitored throughout their growing processes because with each stage, their body is going to react differently. We went over drug absorption, so just understand how drug absorption is affected in children more than it is in adult by the 3 factors: the blood flow at the site of administration, GI function, and then the thin stratum corneum, how that works, distribution, the changes in body composition, how that will affect lipid-soluble drugs, metabolism, particularly the phase 1 enzymes and the phase 2 enzymes, understand how the impact of the CYP450 is, particularly the CYP450-1A2 in children, and then in phase 2, understand how the variability changes And in this one, we discussed mainly know that phase 2 enzymes are responsible for synthesis of water-soluble compounds. We talked about the UGT enzyme system. There are ethnic variabilities as well. Commonly they use medications that are affected by the phase 2 enzymes were acetaminophen, morphine, caffeine, and excretion. Of course, remember, again, as the children grow and mature, things are going to change. And so that's going to change your GFR, your glomerular filtration rates, and will, as renal blood flow changes, then so its excretion is affected. So let's take a break here, and then we'll move on, and we're going to talk about pharmacodynamics.